So let's continue on with our genetics lecture. This is going to look at Mendels Laws, but before that, let's just quickly answer this concept check review. So let's pretend the gene for eyebrows follows a recessive-dominant relationship. The gene for a monobrow is big M. The gene for two separate eyebrows is little m. What phenotype does one have if their genotype is big M, little m, right? So remember that the capital letters are for dominant alleles, the lowercase letter are for recessive alleles. This person has a heterozygous genotype, okay? What's their phenotype? So you should say to yourself that they inherited a dominant allele. They must phenotypically show the dominant trait. So what is the dominant trait? Monobrow. So they have a monobrow, just like this image, okay? So that's the answer. So if you got that correct, good for you. Let's talk about genetic disorders real quick. So there are all kinds of genetic disorders out there. Some of them are recessive disorders and some of them are dominant disorders. Recessive disorders are the most common because what usually happens in nature is that there's this dominant allele that works, right? It allows the organism to survive because it's functional. And that tends to be the dominance, or what we call wild type. Then over time, sometimes those dominant alleles get mutated or they change a little bit, right? And so that causes an alternative version that is usually a recessive phenotype. So in terms of genetic disorders or things like problems, most of them are because of recessive traits. Okay, not all, but most. So if you are a person who is a carrier for a recessive trait, you have to be a heterozygote. So you yourself are phenotypically normal, right? You have a recessive diseased allele, but you are phenotypically normal. So you have no idea, like if you're just, you have no idea by looking at someone that they have a hidden recessive trait, because what they show phenotypically is the dominant allele, even though their other allele might be the diseased allele. However, carriers, right, you can pass on that diseased recessive allele to your offspring. Cystic fibrosis works this way, okay? So carriers for a recessive diseased allele have to be heterozygotes. The term carrier means that you yourself are normal, but you're able to pass it on to your offspring. Dominant disorders are more rare, but they exist. That means that in order to have a disorder, you only need to inherit one copy of that allele, the diseased allele, in order to phenotypically have a disease. So polydactyly and achondroplasia are both examples of this. And actually Huntington's disease is also an example of this, where you inherit just one copy, so just from one of your parents that you inherited a dominant allele. The other one may be dominant, the other one might be recessive, but it doesn't matter because you have a dominant disorder, so you phenotypically have the disorder. This is a picture of achondroplasia. Achondroplasia is a kind of dwarfism. It doesn't allow people to grow taller. There's a problem with their cartilage and their long bones. Okay, so just have an idea that genetic disorders can come in any variety. They can be recessive disorder or dominant disorder. Let's talk about the outline now, letter D, Mendels Laws. So Gregor Mendel, he came up with two laws while he was studying his pea plants. And the first one is the law of segregation, which you already know because we went over meiosis. And all it really talks about is that because your genes are in pairs, right? You have a homologous chromosome set. You have these homologous chromosomes. Those homologs separate during meiosis. So the law of segregation basically just states that the two alleles that you have separate from each other, okay, when in the process of being inheriting or passing on to the next generation. But you know this because of meiosis. Number two, the law of independent assortment. You also know this as well, that in meiosis, the relationship between the chromosomes, they don't rely on each other to assort. they have an independent assortment. So each chromosome lines up along the metaphase plate individually and independent of each other. They don't rely on, they don't say, oh, all of mom's chromosomes are lining up on the left, so I should go on the left, right? It's completely random and independent of each other. So that's all number 2 says, is the law of independent assortment. The alleles of different genes assort independently of one another during gamete formation. Okay, so if you understood meiosis, this is basically just looking at those events of meiosis, but Mendel didn't have that information, so he kind of came up with these laws, looking at the patterns of inheritance. All right, so let's take a look at the Punnet squares that he is. I'm sorry, I didn't flip to this picture. So this is independent assortment. I'm going to talk about these Punnet squares right now, so these will make sense to you. So when Mendel was working his genetics problems, he came up with a way to predict the outcome of the next generation, or at least the possible ratio of the next generation, or the percentages of the different traits in the next generation. And he did so with this Punnet square. So I'm going to try this feature of this to draw in here. So give me a second here. So let's do a cross. So if you're in your outline, we're in Roman number 2, the Punnet squares. This is Mendel's parent cross. So what he did is he crossed 2 purple flowers, so true homozygous purple flowers with 2 homozygous recessive flowers. Okay, so big P, cross little p. So when you do a Punnet square, what you do is you put one parent's genotype on one side of the square and the other parent on the other side. So I'm going to try and draw this. So let's make a big P. This is one parent, right? So this is the purple, big P. And then the white flower, we'll make a little p and a little p. All right, so this is the white parent, white flower parent. This is the purple flower parent. And then when you figure out, like, what are the possible outcomes if I were to mate a white flower with a purple flower, what would I see in the offspring? So you can predict, okay? And this is how you do it. You basically just put the alleles in the box. So in this box, the big P comes here, okay, and then the little p comes here. And in this box, we have the same, right? Big P comes over, and little p. What goes in this box? The little p's go across, the big P goes across, the little p goes down, so big P, little p. In the box, same thing. Big P, little p. Okay. All right, so something erased everything, but hopefully you have written down what was here. And if you asked the question, what are the possible genotypes of the F1 generation? So the F1 generation is the offspring that's here, right? So if you look at each square, what's possible in those offspring? It's just big P, little p. So big P, little p. They're 100% chance of having a heterozygous flower. What are the possible phenotypes of the F1 generation? So if you look at each box would have a big P in it. So the only possible color flower you can get in that cross is purple. Okay, so the answer here would be purple. Let's see if we can answer this. So this is actually true. There's something called the photic sneeze reflex, also called photoptopmosis, autosomal dominant compelling helio-ophthalmic outburst syndrome, or ACHU. Sun sneezing is a condition that causes difficulty to control sneezing in response to numerous stimuli, such as looking at this bright lights. So the allele for sun sneezing, if you look at the sun, it makes you sneeze, it's a dominant big S. No sun sneezing is a lowercase little s. A person is a sun sneezer. What is his genotype? Okay, so you know that a sun sneezer is a dominant trait, a dominant allele. And you can be heterozygous dominant, you could be homozygous dominant, and still be a sun sneezer. So the answer is letter D, both big S, little s, or big S, big S. a homozygous recessive trait, this is not going to be a sun sneezer, right? All right, good. So hopefully you got letter D. And a man who is heterozygous for sun sneezing and a woman who is not a sun sneezer are going to have a child. What's the probability that they will have a sun sneezing child? So this is a Punnett square problem, okay? So let's set that up. So what we're going to do is the problem is set up for you. So big S is sun sneezing, little s is no sun sneezing. And what you're going to do is a man who is heterozygous for sun sneezing. So what is this man's genotype? He's heterozygous, so he's going to be big S, little s, right? And a woman who's not a sun sneezer. So what's her genotype? Right, she's not a sun sneezer, so she has to have 2 recessive alleles. They're going to have a child. What's the probability of them having a sun sneezing child? So you put the genotypes of one parent on one side and the genotypes on the other side. But what this is really representing, when you put a big S here, that's representing this. Remember that the big S, this is an allele on a chromosome, right? So this chromosome went to one gamete. So for a man, this represents one of his sperm got the big S sperm. The little s that goes here represents the other sperm that got the little s allele. For the woman, she's homozygous. So when her cell, that's a diploid cell, undergoes meiosis, one of her eggs gets a big S and the other egg gets one of her eggs gets a little s and the other one of her eggs gets a little s. So what these represent are the gametes of the parent. So when you put the gametes together in any possible combination, right, we're going to have a big S in this box, little s, big S, little s. This is little s, and this is little s, little s. So let's take a look at the chances that they have. So we have 4 boxes. 2 of those boxes contain big S's, right? And then 2 of those boxes contain homozygous recessive. So we would say that this is a 50-50 split. 50% or half of the chances that they're possible offspring, right? Remember, these are possible. They're not going to happen. It's just like this is the probability or the possibility of them having, like this is the ratio of the offspring that's possible for them. So if you look at the boxes, right, so the probability that they'll have a sun sneezing child will be 50% because 2 out of the 4 boxes have a dominant S. That represents the possibility. Will they have a sun sneezing kid? Maybe not. You know, they have a 50% chance. They also have a 50% chance of having a non-sneezing kid. But that's how you would approach these problems and think about these problems, is these are the possible offsprings they have, possible combinations of offsprings, and you know, the boxes represent the percent chances. So we have 4 boxes. That means that we have 25, 25, 25, 25% chances. In this case, we have 2 out of the 4, so it's 50%. So hopefully that makes sense to you. All right, let's move on. The F1 generation cross. So Mendel's F1 generation, he took those F1s, right, the heterozygous purple flowers, and he crossed 2 of those to get the F2 generation. So let's do the cross. So our parent, our heterozygous parent, can have a big P gamete or a little p gamete. The other parent is the same. It could have a big P gamete or a little p gamete. And let's go ahead and fill in the boxes. A little bit different this time. All right. So now, what are the possible genotypes for the F2 generation? Well, we have a lot of different variations. It's possible that we have a big P offspring, right? This box dictates that. It's possible that we have a heterozygous offspring. These 2 boxes are heterozygous. And it's also possible that we have a homozygous recessive offspring, which is indicated by this box. So these are all possible, okay? Phenotypically, what's possible? Purple, 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 white, right? So these three offspring are purple, so both purple offspring are possible, and so are white. So it's possible that you can cross two purple parents and get purple and white offspring. But this is a ratio you need to know, okay? So in your outline, there's a phenotypic ratio for a monohybrid cross and a genotypic ratio for a monohybrid cross. Monohybrid means we're only looking at one. The mono refers to one trait. We're only looking at flower color. And hybrid means that both parents are hybrids. They are heterozygous. Now, in terms of our offspring for every, so let's start with the genotype. There's one big P box. There are two big P, little p boxes are heterozygotes, and then the one homozygous recessive. So the ratio here for genotypes is 1 to 2 to 1 ratio. Okay? It just means that this is the ratio. If you had 1,000 offspring, right, you would find a ratio of 1 to 2 to 1 in their genotypes. In their phenotypes, you have 3 purples for every 1 white flower. All right, that's their phenotypic ratio. Another way of thinking about this is in terms of percentages, because we have 4 boxes. So you can think about it as, well, you have a 75, like if you had a 100, let's just do 100. If you had 100 offspring of these flowers, 75 of them, or around 75, would be purple. And then the next 25 would, and then the rest of the 25 would be white. Okay, so you'd have a ratio of 75% purple to 25% white. You can think of it, the genotype, the same way, right? If you had 100 flower offspring, you would have 25% being homozygous dominant, 50% of them being heterozygous, and 25% of them being homozygous recessive. Okay? All right. I'm having a problem getting out of here.